Good afternoon and welcome back. This is Dr. Sanjeev Pandey and you are listening to StpA Bio. Today we will talk about bacterial chromosomes. Bacterial chromosomes are essential part of all bacteria, all bacterial cells. Uh, we know that the sum total of the genetic material is called the genome of a cell, and bacterial genome comprises of a single chromosome that resides inside the cytoplasm, that floats inside the cytoplasm, along with the ribosomes and other cell organelles, whatever present in a bacterial cell. So bacterial chromosome can only be visualized through electron microscope. Because the bacteria is very, very small, it is difficult to see it under light microscope, and the chromosome is further smaller, and it is only seen under electron microscope. It can be seen under fluorescence microscope if you stain it with some fluorescent dye. Unlike eukaryotes, there is no nuclear membrane around the chromosome, and hence it is referred to as nucleoid rather than nucleus. We know that the eukaryotic cells, they contain a nuclear membrane. They have a proper nuclei. Since prokaryotes don't have a nucleus, a nuclear membrane, a nucleoid, nucleolus, nucleoplasm, and the chromosome itself acts as a nucleus, as a central molecule which controls the entire function of the cell. So that's why these cells, the bacterial cells, are called prokaryotes, because they don't have a true sense of nuclei bound by the nuclear membrane. So this is called nucleoid. It is also called genophore because it bears gene. Phore means bearer and geno means genes. So gene-bearing structure. So that all chromosomes are gene-bearing structure, of course. So now let us see how it is organized inside the bacterial cell. So here is the bacterium and the electron microscopic structure of it. You can see that inside the bacterial cell, this is highly supercoiled, highly condensed genetic material, that is the chromosome of this cell. This is an E. coli bacterium, Escherichia coli, the typical bacterium which has been studied the most. If you take out this chromosome outside and if you spread it completely, it takes a circular shape like this. And it is called CCC DNA. That means covalently closed circular DNA. It is covalently closed circular DNA. There is no end of it. There is no centromere. There is no nucleolar organizer region. There is no telomere. So the question of telomere and the problem of the size reduction in every round of replication is avoided by this circularization of the chromosome. The thing is that this completely spread chromosome, it has a size of about 350 micrometer in diameter, and the cell is 1 to 5 micrometer in diameter and length, in breadth and length. So how can such a bigger molecule can be accommodated inside this very, very smaller compartment? For that, this chromosome has to go compaction, has to be packaged in a way so that it becomes smaller and can be fit into the bacterial cell. So the first level of packaging that occurs within this chromosome circular molecule is these loop-like structures that are formed with the central proteinaceous and RNA core. This DNA material is stacked in the middle, and the loops radiate from the center. There are many such loops, maybe 30 to 300 loops at different stages of cell division, cell growth rather, not cell division. When they are in logarithmic phase or in stationary phase, the thing is different. So there are many loops coming out from a center. We will see that in one of the electron microscopic micrographs. So at the first level of compaction, it reduces from 350 micrometer to 30 micrometer size. which is also bigger compared to the bacterial size. So it has to go further compaction. How it occurs? Each of the loops are further supercoiled. They are highly coiled within themselves, and it was proved by coiling within themselves, the size reduces to about 2 micrometer, and now it can be fit if it further goes a third level of coiling, which occurs because this material is present in a helical way inside the cell, and it is elongated like this, so it can fit inside a bacterial cell. It takes the 1/3 volume of this cell, of course. So these loops are consisted of supercoils. It can be if you treat this chromosome with a with a dilute concentration of DNAs, means if you partially digest it with DNAs, then what happens is some of the loops get opened like this. Because this is a double-stranded, each of these loops are double-stranded DNA and supercoil. So if you cleave one of the DNA strands, then what happens? Then the supercoil is relaxed and the loops become open like this. There is a nick in the DNA, in one of the DNA strands, not both. Here also there must be a nick inside one of the DNA strands so that it opens up like this. So each of the loops are highly supercoiled. It is proved by treating this chromosome with the dilute concentration or mild DNAs. Now again, if you treat this chromosome at this level, it is difficult to isolate and get this kind of chromosome from a bacterial cell. We will see that how it is done. So if you digest it with RNAs, partially digested, then what will happen? These loops get opened like here. The supercoils are not relaxed, but these loops this part is clipped. That means this part is held by the RNA. Each of these loops are held by RNA. So if you treat it with a if you partially digest it, you will get some of the loops open. If you digest it completely, then in the next diagram we will see, if you can increase the concentration of RNAs, then what happens? The entire chromosome, it opens up like a circular molecule like this. This is a picture taken from Arthur Kornberg's book, The DNA Synthesis, where he has shown that the RNA molecules, that single-stranded RNA, they held these loops, supercoiled, radiating looped domains in this manner. They hybridize at each of these central portion of these foldings of the DNA. They held here, and the RNase A, it clips this RNA, and that's why these two loops get opened up. These loops get opened up. If you increase the DNase concentration, each of these single loops get opened, and if you increase the RNase concentration, the entire chromosome from the center, it gets clipped and it becomes open. If you treat both DNase and RNase, it will be like this. So this proves that these looped domains are further supercoiled. They form secondary structures, and they are held by in the center by some RNA molecules also, along with the proteins that we will talk about. So this slide shows that different microscopic photographs taken from bacterial chromosome. This one is a fluorescence microscopy. The bacterial cells have been stained with StpA, and then they have been observed on fluorescence microscopy. You can see that these chromosomes, the outline of the cell is not visible here, only the chromosome is visible. You can see the condensed material. It is getting separated as it seems like that, but it is not separated actually. Or might be in this case the cell is going cell division, that's why it looks like this. So this picture was already there. If you take out the chromosome, if you prepare it in a very gentle manner, and that part is very difficult to get this chromosome out and spread it in the gentle without disrupting it, it's very, very difficult, though it can be done. That if you take it out, if you digest the cell mildly and then then treat the chromosome. Chromosome still remains in a very condensed state, and if you do an ultracentrifugation with this density gradient centrifugation rather, using sucrose gradient, then you get the band of the DNA on an electron microscopic grid, and you stain with uranyl acetate, and then you observe under the transmission electron microscope. It gives a picture like this. This picture again shows that the DNA, the chromosome, is attached at the center by some material. These are proteinaceous material as well as some RNA, and these loops are radiating from the center outside, and they're highly supercoiled also. So this gives the same picture which in the earlier slide we discussed. And if you draw the diagram from this, it looks like this, that these loops are highly supercoiled and they're held at different places by different kinds of proteins. These are proteins of different kinds. The RNA, polymerase, HNS protein, histone-like nucleoid structural proteins, transcription factors. These are transcription factors because transcription is also going on. There are topoisomerases, there are IHF proteins, there are HU proteins, and there are StpA protein. There are a number of proteins. There are FIS protein. We will talk about them in the next slides. There is another model about this chromosome, that this in certain state of the cell, the chromosome takes a shape inside the cell. It takes a axial elongated shape, and this model is called bottle brush model. This model is being called looped domain model, and this one is called bottle brush model. Both the models are probably right at some stage of cell growth. This is outside the cell when it is prepared on the slide, and this is inside the cell in the in vivo condition. So it may be that so that in vivo condition the chromosome, the loop domain structure remains in an elongated fashion like this, and it forms a bottle brush kind of structure. So now we talk about different proteins associated with the chromosome. There is a nucleoid-associated proteins. These nucleoid-associated proteins they help in the organization of the bacterial chromosome, and the DNA binding protein, of course. Nucleoid-associated protein bind non-specifically across the bacterial genomes, and they help in wrapping, bending, and bridging of the DNA or of the chromosome. The local action of nucleoid-associated proteins ultimately influences the global organization of the chromosome, and in many cases its transcriptional pattern is also controlled by these nucleoid-associated proteins. In E. coli, the HNS, this histone-like protein, histone nucleoid structural protein, is a small protein that can dimerize and can bridge DNA, bringing loci close at close physical proximity. We will see that there are other proteins which also help in holding the DNA. The holding the DNA's secondary structure in different ways. You can see this SMC protein, structure maintenance of chromosome protein. This protein is a dimeric structure. It heads the chromosome. It forms a bridge like this, and it heads the secondary structure in this manner. The FIS protein, they form a dimer, and they also help to bend the chromosome like this. The integration host factor protein also help to make a dimer. They are not in a dimeric form; they are monomeric form. However, they help to bring a curvature, a bending of the chromosome like this. The HNS proteins, they are usually in the form of a dimeric structure. So they actually bridge between the two double-stranded double helix that is going on. They tag between them, and they bridge between them, so that the two DNA strands, they come close to, not two strands, but rather the double-stranded helix, they come close to each other, forming a secondary structure, a kind of a secondary structure. This is the supercoiled structure held by the HU protein. The HU protein might form, there are two types of HU protein, HU alpha and HU beta. We will talk about them a little bit. Here is it, the role of HU protein is very, very significant in chromosome condenses. Prokaryotic cells contain a set of proteins that bind the DNA and are associated with the nucleoid. This set of proteins are called HU. Among these proteins, not the only HU. There are others that we talked about: the HNS, the StpA, the SMC, IHF. All of them. These are the set of proteins. Among these, the protein HU has been characterized the most, and it's most important as far as the chromosomal organization is concerned. For example, the lack of HU, you can see in this kind of mutants, HUPA and HUB mutants, it is being seen that the nucleoid becomes decondensed. In mutants of U protein, HUPA and HUB, the nucleoid doesn't condense in a manner that we are talking about. So that means it helps in the condensation of the DNA. In E. coli, HU exists predominantly as a 20 kilodalton heterodimer, HU alpha and HU beta, composed of two subunits, that is HU alpha and HU beta. These are encoded, of course, by HUPA and HUBB genes because their mutation causes the decondensation of the nucleoids. Now, while HU was initially characterized as the bacterial equivalents of histone protein, it was initially said that these HU are the equivalents of histone proteins of eukaryotic system. More recent studies have revealed that the main role of HU is in a number of cellular processes is that of a DNA architectural protein that bends the DNA in a non-specific, non-sequence specific manner. Thus, the HU appears to be more similar in function to HMG proteins of the eukaryotes than it is to histone proteins. So, more closer to histone proteins are the HNS protein, IHF protein, and it was also told that HU proteins are also closer to histones, but they seem to be more closer in characteristic to HMG proteins of eukaryotes. And how does the HU actually interact with the DNA? The structure has been revealed in Anabina that the HU, if you co-crystallize the HU protein with the DNA, it reveals that they have a conserved proline within their structure, which intercalates between the bases of the DNA in the minor groups to induce pronounced kinks or bends in the double helix. In this way, these HU proteins actually interact with the bacterial chromosome. Now, there is another protein called PAR-B that also associates with the bacterial chromosome, particularly at the time of when the cell is dividing and when the chromosome is also getting separated. So, these PAR-B proteins bind with a sequence called PAR-S on the chromosome, which is located very, very closer to the ORI site, origin of replication site. So you can see this is the binding site of PAR-B proteins, and this structural diagram is showing that the PAR-B proteins has been fused with the GFP, green fluorescent protein, just to see their localization, where they are localized. So they are found to be localized on these two parts of the chromosome, which are the origin of replication sites, and it is assumed that this PAR-B protein interacts with the cell membrane when the cell is getting separated on both the poles, and they separate out, they stretch out the DNA so that both the daughter cell get each of them get a chromosome when the chromosome is also dividing at the same time. It has to be distributed in both the daughter cells equally. So that is that part is controlled by the PAR-B protein, that it gets separated and distributed properly. So now let us say that the number of chromosomes that in the first slide we talked that each of the bacterial species or strain, they contain a single chromosome. The bacterial genome comprises of only one chromosome, whereas in eukaryotic system we see that there are many chromosomes. In different eukaryotes, there are many chromosomes. But in bacteria, we see only there is one chromosome in most of the bacteria. However, there are exceptions. In Vibrio cholerae, you find 2 chromosomes. In Paracoccus denitrificans, you find 2 chromosomes. In Rhodobacter spheroides, also you find 2 chromosomes. In Agrobacterium tumefaciens, you get 2 chromosomes, one circular and one linear, interestingly. So linear chromosomes are also found in bacteria, bacterial system. These are exceptions, of course. The linear chromosomes are also the exceptions. So because all the chromosomes in bacterial systems are covalently close circular, but you get linear chromosomes in Borrelia burgdorferi, in Streptomyces silicolor. How do these look like? These linear chromosomes, they are more or less like it looks like a transposon. It looks like a TN element, apparently, because it contains on the both sides, both terminal inverted repeats. And the other genes are present inside them. It is very, very large because it's the whole chromosome that contains all the genes required for a cell to qualify or become a cell. These terminal inverted repeats, these structures, they help the chromosome to circularize like this. These two ends can hybridize with each other, and they are held by certain proteins also. So basically it forms a circular structure which is not covalently closed, rather it is a non-covalent circular molecule. So in case of Borrelia burgdorferi and Streptomyces, the DNA, the chromosome is not circular. It is circular, but it is not covalently closed circular. It is a linear DNA, basically. So another thing which helps in the compaction of the bacterial chromosome are the repetitive extragenic palindromic elements, the REP elements. These elements, these are palindromic elements, and they have been discovered in Enterobacteriaceae family, in Enterobacter, in Pseudomonas putida also. And they are found to be there are 30 to 50 base pair long repetitive elements, and they are in palindromic sequences, so they can hybridize with each other. And you can find about 300 to this will be 1,000, not 1,000 copies of these elements in a genome. It varies among different species. However, the number varies from 300 to 1,000 copies in bacteria to bacteria. And how do they actually help in compaction? They form this kind of structures, stem loop type of structures. This kind of structure they form, and in this manner they help to reduce the volume of the chromosome. So it further adds into the compaction and into the packaging of the chromosome. So I think this was good enough for the bacterial chromosome. We talked about it in details. If anything has been remained, you can discuss it in the comment box. You can put your questions over there, and I will try to answer them. And if anything has remained, or if you want to add anything in this information, you are welcome to add that in the comment box. Thank you very much for your silent listening.